All right, Fire in a Bottle videos are back. I hope you all survived your holidays. If you're new to the channel, I'm expecting a bunch of new people this week due to my appearance on the Paul Saladino MD podcast. That should be out tomorrow, I believe. So that's a fun audio recording with Paul and I. Go check that out. I've been discussing a lot something called PPAR alpha, which is a nuclear receptor. Nuclear receptors are proteins that can monitor for external signals, and they respond by turning on different genes within the cells. They're kind of master controllers of multicellular animals and our metabolism and how it all works. Things like estrogen, steroid hormones, vitamin D, thyroid hormones, vitamin A, the PPARs, and the ARA hydrocarbon receptor. Those responses are all controlled by nuclear receptors. And I listened to a bunch of podcasts about ancient biology, and I read a bunch of papers about ancient biology. So I'm going to spend 5 minutes in the beginning giving you a little background about, kind of how we got here, right? I've been discussing what exactly does PPAR alpha do. PPAR alpha has been called the master regulator of lipid metabolism. I don't believe that's really what it is. I believe that PPAR alpha started off as a detoxification enzyme, and interestingly, it is also very involved in our response to hypoxia, and that means low oxygen environments. This first slide has two different papers on it. We're going to go into a really interesting paper and a really interesting scenario in humans involving hypoxia and diabetes risk, low oxygen. So stick around for that, but I'm going to give you 5 minutes of history. How did we get here? Why are these nuclear receptors so involved in low oxygen environments? We're going to start in C. elegans, which are nematodes. Nematodes are the most plentiful animals on Earth. They separated from humans about 600 million years ago, maybe further. If you put them, this is low oxygen here. If you give them a low oxygen environment, the expression of PPAR alpha goes up. In the low oxygen environment, when PPAR alpha goes up, the worms accumulate more fat. So this is a different study, but this is anoxia, low oxygen, and you can see the fat build up as well as the PPAR alpha. And so this is breaking news. You can see this is from July of 2023. I almost said July of this year, but it's 2024 now. It says, as long as 800 million years ago, multicellular organisms began to appear in all of Earth's oceans, including sea sponges. And this was thought to be driven by increasing oxygen concentrations. But these guys did this study and they measured oxygen concentrations, and they found that way back then, the oxygen had not really increased. Indeed, levels remained 5 to 10 times lower than today. Which is roughly how much oxygen there is at twice the height of Mount Everest. And so multicellular life began in a relatively low oxygen environment. This is a cool photo of a sponge that's 600 million years old. This is a very ancient creature. Sponges are believed to be the oldest, the most ancient form of life that still exists on the planet. Sponges are the oldest extant animal group. The first fossil demosponge records date from 750 million years ago, and we know that sponges are animals because they have diploid multicellularity, which means they are multicellular animals, and they have 2 pairs of chromosomes in each cell, just like you do. They go through the process of meiosis, which is the division of our sexual cells like eggs and sperm, as well as oogenesis and spermatogenesis, just like you do. And these characteristics clearly differentiate sponges and other metazoans, which means animals, from their assumed closest relatives, the choanoflagellates. And this is a choanoflagellate. This thing is the closest thing we know of to animals. You can see where it is on the family tree here. It's right. These are animals. There's humans. Here's sponges. And right behind them are the choanoflagellates. And interestingly, in these choanoflagellates, there are no receptors or ligands were identified from the nuclear hormone receptors. This means they don't have PPAR alpha. They don't have a retinoid receptor, a vitamin A receptor. They don't have a vitamin D receptor. These things are not animals. They don't have nuclear receptors. The sponge, on the other hand, this is in the marine sponge. This is the sequence of retinoid X receptor, which is their vitamin A receptor. This is a protein sequence. So proteins are made of different amino acids. It's like a language, and this is a sponge protein sequence, and this one here is a human sequence, and this is C. elegans, and that's Drosophila melanogaster. That's a fruit fly. Um, and you can see that all these black lines are essentially perfectly conserved. So that means that this is you can see the zinc finger module. That's like the money part of the nuclear hormone receptor RXR. That's where it binds DNA. And so you can see that these things are in perfect sequence alignment, despite the fact that we presumably, our last common ancestor of sponges was 800 million years ago, okay? And these sections of this protein are still in 100% conservation between sponges and humans, because once something works in biology, the functional parts of it are perfectly preserved for incredibly long periods of time. This is potentially one of the first chordate fossils. This is found in a 525 million year old rock in China. A chordate is a thing with a spinal cord. This thing wouldn't have had a skeleton yet. It doesn't have a jaw. It doesn't have a lot of things that modern animals have, but this is essentially our great grandmother. We evolved from this thing that came out of 250 million years of animalian evolution in low oxygen oceans. You can see it looks like a small fish. There's animals alive today that are very much like this thing. They're called Amphioxus. And what's really interesting about this is Amphioxus encodes 33 nuclear receptors, including PPAR. So there were 250 million years of evolution of these first simple multicellular animals like sponges, which in low oxygen seas, these animals had to deal with changing ocean conditions. When tides came in, you might be in higher oxygenated water, you might be in lower oxygenated water. If you were growing near a bunch of algae, you might have more oxygen if it was sunny and the algae were doing photosynthesis and producing oxygen. And so there's all these different microenvironments. These are animals. They have mitochondria. They're capable of using oxygen to generate a ton of ATP from their fuel. However, they need to be prepared for low oxygen situations, which would have happened all of the time. And there was 250 million years of this kind of evolution before we have something that looks like something with a skeleton, which would be this Amphioxus creature or the one in the rock in China, take your pick. And so you might think, okay, what does that have to do with me now? I'm a modern human. I live in an oxygenated environment, but humans actually have to deal with hypoxia or low oxygen all the time. One example is if you have a heart attack or a stroke, you cut off blood flow to certain tissues. If you have pneumonia, you can't absorb as much oxygen through your lungs. Obviously, if you drown or if you asphyxiate, that's a low oxygen circumstance. Sleep apnea is very common. This is when you lay down, especially if you lay down on your back to sleep, your airway actually closes many times per night. It's best not to sleep on your back, sleep on your stomach. You'll get more oxygen that way. There is a lot of examples that particularly adipose cells in obesity are hypoxic, and that's causing problems. And cancer cells, tumors, are very dense groups of cells, and they don't always have enough blood flow. They can't build enough blood vessels. The tumor's growing so fast, they can't supply enough blood to the tumor, and so the tumors become hypoxic or anoxic. And so I teased this on that first slide, but what this shows is that in periods of hypoxia, the PPAR alpha, which is called NHR49 in these C. elegans, PPAR alpha is massively increased. And if these are worms that don't have PPAR alpha, and if you put them in a low oxygen environment, they fail to grow and they fail to live their life cycle if they don't have PPAR alpha. And so this shows that PPAR alpha, the presumed master regulator of fat metabolism, is for some reason crucial to surviving low oxygen conditions. And this was also on that first slide, but this just shows that if you restrict oxygen from a C. elegans worm, they get fat. This is a red dye that detects fat, and you can see this is the width of the worm, right? Their waistlines increase. All you have to do is restrict oxygen. And this is 3T3L1 cells. These are fat cells taken from mice, okay? So now we're up to mammals. This is hundreds of millions of years of evolution later. If you grow them at normal atmospheric oxygen, they accumulate this much fat. That's 20%. If you decrease that oxygen down to 4%, they accumulate about 3 or 4 times as much fat. This is the size of the fat cells. And this is sort of a weird graph. The larger fat cells are this way. And so these more than 50 micrometer fat cells are much more common in 4% oxygen than they are with 20% oxygen. And that brings us to humans. And so this study is called Obstructive Sleep Apnea is Independently Associated with Insulin Resistance. Stepwise multiple linear regression analysis showed that obesity was the major determinant of insulin resistance, but sleep-disordered breathing parameters were also independent determinants of insulin resistance. This association between OSA and insulin resistance was seen in both obese and non-obese subjects. They looked at the minimum oxygen saturation of the blood of people with sleep apnea, and they divided them into, as you go to group 4, you have the highest amount of sleep apnea, and you can see these people with intense sleep apnea, their minimum oxygen saturation goes all the way down to 64%. And so that's a really anoxic way to spend your night. And you can see the insulin difference. The insulin in those people is more than twice. If you have severe sleep apnea, your insulin more than doubles on average, and your insulin resistance score goes from a 1.6 all the way up to 4.0. And so if you have sleep apnea, regardless of whether you are obese, you are going to be very insulin resistant. These are actually human liver cells grown in tissue culture, and this is aerobic, so these have access to normal air, and these are hypoxic. You can see under hypoxia. And so you can see the lactate levels when you remove the oxygen increases by 6-fold. And the ratio of lactate to pyruvate increases by 5-fold. And so if you take oxygen away from tissues, they produce a lot more lactate. Furthermore, in humans, this is a study of obese subjects versus normal controls. This is obese humans in the morning, fasted, at rest. And this is how much lactate is being released by their adipose tissue. In the obese subject, you saw about twice the lactate release. Obese humans' adipose tissue is producing more lactate in the fasted state. Why does that happen? Well, this is a very high-level overview of how glucose metabolism works. So glucose comes into the cell and it goes through a process called glycolysis. Glycolysis does not require oxygen, and 1 molecule of glucose turns into 2 molecules of pyruvate. And what you get out of that is you get 2 ATP, and ATP is good. You can run a cell on ATP, and you convert 2 molecules of NAD plus to 2 molecules of NADH. And if there's lots of oxygen available, those pyruvate will go into the mitochondria. They'll go through the Krebs cycle. They'll crank out a lot more NADH, and those NADHs give their electrons to the electron transport chain, and that is where oxygen is burned. Oxygen is converted to water by the electron transport chain, and that gives us NAD plus back. And NAD plus allows us to do glycolysis, and it allows us to run the Krebs cycle. And so you need oxygen to convert NADH back to NAD plus, and that happens in the mitochondria and the electron transport chain. Got it? Now, if you have a hypoxic situation, what happens is you can't run this electron transport chain anymore. And so NADH is going to build up, right? The rate of this reaction is going to slow down, and you're going to have a pileup of NADH. And so we already saw that in a hypoxic situation, PPAR alpha is increased. And what PPAR alpha does is it actually blocks this enzyme, pyruvate dehydrogenase, which is the thing that allows the pyruvate to go into the Krebs cycle to be burned as fuels. So what cells do about this is, of course, they can't do oxidative phosphorylation in the mitochondria anymore. And so they divert this pyruvate to lactate. And when you convert pyruvate to lactate, you actually take this pair of electrons, you stick it back onto the pyruvate to make it to lactate. That gives you NAD plus back. And you can keep doing glycolysis and you can keep making pyruvate, and the lactate just simply gets exported. When you saw in those obese humans, when you saw lactose increasing as the blood moved through the adipose tissue, those adipose cells were doing glycolysis to get these 2-ATP, and then they were fermenting the pyruvate back to lactate, and they were exporting it. And so you can use fermentation to get rid of extra pairs of electrons that build up as NADH. And that is why all things ferment. So whenever you see a fermentation process, those fermentation products are essentially wasted energy. When you see fermentation, it means things don't have oxygen. They can't break them all the way down to carbon dioxide and water. And so instead of getting all of the ATP that comes from the electron transport chain, they're just dumping things like lactate, which could be burned, but are not going to be burned in this case. When you think about sauerkraut, the bugs in the sauerkraut are eating the sugar, and there's no oxygen in a vat of sauerkraut, and they can't do this oxidative phosphorylation. And so they ferment the pyruvate that they get from that sugar into lactate, and that becomes lactic acid. And the lactic acid is that sour flavor that you get when you eat sauerkraut. So another name for this, when you can't do oxidative phosphorylation, what happens is NADH builds up, NAD goes down, and this is called pseudohypoxia. And pseudohypoxia is involved in the pathogenesis of type 2 diabetes. It's well known that in diabetes and in obesity, you're in this mode called pseudohypoxia. And what I like about this study is they got the sequence of events exactly right. You start with, they say diabetic hyperglycemia, and I added to the list activated PPAR alpha and aryl hydrocarbon receptor, and also lots of dietary unsaturated fats can give you or I'll even add to that, if you get into a torpid metabolism, if you're eating a lot of branched chain amino acids, that can also give you this NADH to NAD plus redox imbalance, which they say can lead to pseudohypoxia, which can lead to reductive stress. I put these into a box because in my mind, these are all more or less the same thing. And so you do these bad things, you get into pseudohypoxia. And it's this pseudohypoxia or the NADH, the reduced things, the reductive stress, all of that ultimately leads to oxidative stress and then cell death and tissue dysfunction. And like I say, I just like that they got the order of events right. It starts with reductive stress. It starts with lack of oxygen. Lack of oxygen is ultimately what causes oxidative stress. And that seems counterintuitive. That seems ironic, but that's the reality. And so that brings us to the paper that I really want to focus on. So this paper is fairly remarkable. I think it came out in 2019. It's remarkable to me that the world doesn't talk more about this paper because I think this is such an important topic. And what they point out in this paper is that I talk a lot about desaturase enzymes. I talk about SCD1. SCD1 is incredibly implicated in obesity, and these other two desaturase enzymes called D6D and D5D are sort of co-equally implicated in progression of obesity and diabetes. And these are all desaturase enzymes. What they do is, they provide a mechanism for glycolytic NAD plus recycling. And what they say, and this is the key phrase, it's analogous to lactate fermentation. And so what they're saying is that process of when you are converting pyruvate back to lactate so that you can get NAD plus back and keep doing glycolysis, the same thing happens when you desaturate fats. And so what they show here is there's this enzyme called SLC16A11. You don't have to memorize it. It brings pyruvate into cells. And so when cells get into pseudohypoxia, they're not getting enough oxygen, they have too much NADH, they can bring in pyruvate, and they can use that pyruvate to ferment to lactate, and they can regenerate NAD+. Their other option, as this paper points out, is you can take PUFA, polyunsaturated fats, and you can convert it into HUFA, which is highly unsaturated fats. And so this is the difference between linoleic acid, which is 18,2, which means it has 2 double bonds, and arachidonic acid, which has 4 double bonds. So in this schematic, arachidonic acid would be HUFA. But either one, this is the sort of the main pathway that normally happens in cells. Cells export pyruvate and lactate, and they're constantly bringing it in and out, and different tissues can use circulating pyruvate and lactate in the blood to balance their own kind of redox status. And so this is how normal cells do it. You can bring in pyruvate If you get into a situation where you're pseudohypoxic as a cell, you can bring in pyruvate and you can use it to regenerate some NAD plus so that you can keep running your metabolism. However, there's this mutation in human type 2 diabetics where this enzyme or this transporter that brings in pyruvate does not work as well. And so if this thing isn't working, what happens is NAD plus availability goes down. And if this pyruvate transporter is not working, this is strongly associated with development of diabetes in humans. So if humans can't bring in pyruvate to regenerate NAD+, they will become diabetic, or they have a very strong chance of becoming diabetic, and NAD+ levels drop. And so just to go back to our little map here, I know it's getting a little busy. This paper is suggesting an alternate. Instead of taking pyruvate and using it to regenerate NAD+ and ejecting it, what we can do, and this all happens due to PPAR alpha, and this is why PPAR alpha is upregulated during hypoxia. PPAR alpha controls delta 6 desaturase and delta 5 desaturase, which are the things that ferment the polyunsaturated fat into highly unsaturated fat like arachidonic acid. And when you do that, you get NAD plus back. And so PPAR alpha is, first off, it's blocking the entrance of pyruvate into the Krebs cycle or the TCA cycle. Secondly, what it's doing is it is causing you to ferment your polyunsaturated fats into highly unsaturated fats. And that's giving you NAD plus back. And that is allowing you to continue to do glycolysis and continue to bring in glucose. And you still, just like with the lactate, you still have to get rid of these things. PPAR alpha also controls cytochrome P450s, which further oxidize the arachidonic acid to things like 5-HEAT, which are strongly associated with obesity and diabetes. And it allows you to get the 5-HEAT out of the cells by a process called glucuronidation, which is a detoxification pathway controlled by PPAR alpha, and you can actually urinate out glucuronidated 5-HEAT and other oxylipins. So one of the reasons your body is making these oxylipins, which are associated with obesity and diabetes, is that PPAR alpha, through this fermentation pathway, allows your cells to get out of hypoxia, allows your cells to get out of pseudohypoxia. And that is actually why PPAR alpha is causing us to oxidize polyunsaturated fat. And that study showed that if you block the enzyme that converts pyruvate to lactate, that fermentation of these polyunsaturated fats increases. And so we know for sure that in hypoxia, fermentation of polyunsaturated fats to oxylipins helps us continue to do glycolysis, and that glycolysis pathway is exactly what we see in obesity. And so again, this is from that glycolytic recycling paper, and I was very much on the fence about whether or not I wanted to show this graph because it's kind of insane, but it's actually really interesting. So I'm gonna lead you through it. It's not as bad as it looks at first glance. And so again, I just want to introduce you to the concept. So the problem in these humans is they have a defect in this transporter that allows them to bring pyruvate into their cells, and bringing pyruvate into your cells, of course, allows you to fix potential redox imbalances. So in any given cell or any given tissue, if a cell or a tissue gets in trouble and it gets into pseudohypoxia, it can bring in pyruvate and it can kind of rebalance itself. However, if you have a defect in this transporter, then your only option is to fix your cytosolic redox balance by fermenting PUFA to HUFA, by fermenting linoleic acid to arachidonic acid, and also by fermenting saturated fats to monounsaturated fats using an enzyme called SCD1. So you can convert stearic acid to oleic acid. And so these are in actual humans. These are in actual humans who have this specific defect in this pyruvate transporter, which leads to diabetes. So what we're looking at here is these are triglycerides. So fat is in triglycerides, right? So usually we talk about things like oleic acid, which have 18 carbons, right? and 1 double bond. And so if we look at 54, right, that would be a triglyceride. So a triglyceride is just 3 of those fatty acids connected to a glycerol. So if we talk about 54-1, that means that you have 3 18 carbon fats. So that's probably something like, and there's only a single double bond in 54-1. So that would be like stearic acid, oleic acid, right? That's 18, 18, 18 equals 54, and there's a single double bond. So that's a 54-1 triglyceride, okay? Let's look at 52-0, because that one is almost certainly going to be 2 stearic acids and a palmitic acid, right? It's an 18 carbon and an 18 carbon and a 16 carbon. You add that up, you get 52. You get 0 double bonds. That means they're all saturated. So they're dividing the amount that you find in that in a person with this defect, this person that's in pseudohypoxia versus a normal person. And what they find is that this 54-2 is less common if you're in pseudohypoxia. Why is it less common? It's less common because the person in pseudohypoxia who can't pull in pyruvate has no choice but to fix their redox balance by desaturating their fat. And the other thing you'll notice is that when you think about something like arachidonic acid, so linoleic acid is 18 carbons long. It has 2 unsaturated bonds. When it goes to arachidonic acid, it becomes 20 carbons long, and now it has 4 unsaturated bonds. And so the other thing that happens in this process is the fats get elongated. So this is the line that's 1.0. So that means the same amount in someone with pseudohypoxia or with a defect in pyruvate transport as someone who's normal. And so these short chain fats, the people with pseudohypoxia have less of. And these long chain fats, the people with pseudohypoxia tend to have more of. So I picked this is totally arbitrary. I picked 52-2. So one combination that could make 52-2, and this is a bunch of different things that can be 52-2. But one example would be a palmitic acid, that's 16-0, stearic acid, that's 18-0, linoleic acid, that's 18-2. Add it all up, you get 52-2. Great. Okay. As these things get unsaturated, as they get fermented, because the cell needs to regenerate cytoplasmic NAD+, what it does is it converts the palmitic acid to palmitoleic acid, the stearic acid to oleic acid, and the linoleic acid gets both desaturated and elongated to make arachidonic acid. And so this 52-2 has the same starting fats as 54-6, right? And so you see that in the people with pseudohypoxia, you see a lot more things like 54-6 because the fats are getting desaturated, they're getting elongated. Okay, sorry, that was a long, nerdy description of a cool graph. But now here's why it's interesting to understand it. They then took this bad version of the gene and they incubated these. This red is the bad version of the gene incubated in a normal medium, and the blue is the bad version of the gene incubated with a lot of pyruvate in the solution. And so the activity level of this thing isn't 0, right? It's something. So if you give it a lot of pyruvate, it can regenerate NAD+, and it doesn't have to continue fermenting polyunsaturated fats. Like I say, this is in cell culture, but the cells with the bad version of the gene with tons of pyruvate tend to stay below the line, even though they have the bad copy of the gene. If you flood it with enough pyruvate, that will allow you to regenerate NAD+. It will get you out of pseudohypoxia, and you will have more saturated fat. So pyruvate actually increases the saturation levels of your fat. This is like I say, is in cell culture. It's not the same as a live animal, although it may work the same in an actual human being. This is just to show that this same process is happening in obese humans. And so you can see that as humans move from lean to overweight to obese, the amount of linoleic acid decreases steadily, and the amount of DGLA, which has a third, so linoleic acid has 2 unsaturated bonds, DGLA has 3 unsaturated bonds, and that goes from 2.08 to 2.64 to 2.98. Arachidonic acid goes from 9.8 in lean to 10.38 in overweight humans. And these weird, extra long, very unsaturated omega-6 fats that we don't even hear about, those are also go from small amounts in lean humans to more in overweight to significantly more in obese humans. And so we just see again, obese humans are producing a lot of lactate in their adipose tissue, and they are fermenting linoleic acid to highly unsaturated fats to help regenerate some NAD+. If people ask me if I want to take one supplement, what should it be? I typically will say either alpha lipoic acid, or I'll say the SEA that I sell at fireinaBottle.net/shop. But alpha lipoic acid is very, very good. And this is in obese patients with type 2 diabetes. This is an actual walking around human beings. This is a control group here. These are obese diabetic humans. You can see they produce massively more lactate after a glucose challenge. What alpha lipoic acid does is it oxidizes NADH to NAD+. So alpha lipoic acid can become the terminal electron acceptor in your mitochondria. It can effectively take the place of oxygen and help to eliminate pseudohypoxia. And so when you give alpha lipoic acid to obese humans with diabetes, the amount of lactate that they produce after a glucose challenge goes way, way down. If you've watched my videos, you know I'm a huge fan of oxidants. The two best oxidants, the two simplest with the fewest side effects that we know of, are pyruvate, which oxidizes cytosolic NADH, and alpha lipoic acid. I prefer R-alpha lipoic acid. It costs a little more, but it has fewer side effects. R-ALA. The oxidants are there to get you out of pseudohypoxia, to get you to stop fermenting your PUFA, to get you to stop converting stearic acid to oleic acid, to help you resaturate, help you stop producing lactate. Both of these things, this is pyruvate supplementation. It's been tested a bunch of times. These are not great studies, but even in the handful of not great studies, there is pyruvate supplementation statistically causes people to lose weight, not a ton of weight, but it's one lever that we can pull that does seem to actually have an effect in humans. Same thing with alpha lipoic acid. Alpha lipoic acid has been tested a lot. Overall, it does seem to have some beneficial effect on weight loss. Again, by itself, it's not a panacea, but each of these things are levers that we can pull to help us in our journeys. And so, as usual, I've thrown an incredible amount of information at you. My hope is that if you watch enough of these videos, you'll start to learn the language over time. It's like submersing yourself in any language. Hopefully, if you watch a few of these, you'll start to get the hang of it. But in this video, what I showed you is that we have this family of nuclear receptors, including PPAR alpha, and these nuclear receptors have been subjected to literally 800 million years of evolution. They show up in the very first multicellular organisms. They're not in our most closely related single-celled organisms. And so definitionally, when things became animals, that is when the first nuclear receptors show up. And there was 250 million years of nuclear receptor evolution in a low oxygen environment before we have the first chordate, the first thing with a spinal cord. In low oxygen conditions, PPAR alpha causes the fermentation of PUFA, of polyunsaturated fat, into highly unsaturated fats. And this fermentation, this pseudohypoxia, drives the production of oxylipins, the oxidized lipids that are strongly associated with development of obesity and development of diabetes. Pseudohypoxia can be defined as a high ratio of NADH to NAD+. It is caused by lack of oxygen. I showed that in that tissue culture study with liver cells, and they remove the oxygen for an hour, and the ratio and the amount of lactate goes up 6-fold. You don't want to be pseudo-hypoxic, so don't sleep on your back. Sleep on your stomach, sleep on your side. Do anything but sleep on your back, especially if you're obese, especially if you have metabolic syndrome. You need that oxygen. And then lastly, consider using the oxidants. Like I say, RALA is my absolute favorite, but pyruvate's an interesting one. Calcium pyruvate you can get. It's cheap. People take up to 6 grams a day, or some of the studies with pyruvate, they use 35 grams a day. That's a lot of calcium. That's a lot of pyruvate. I don't recommend taking that much. ALA, I've been taking 240 milligram capsules of RALA twice a day with meals, and about 6 grams of pyruvate recently. That's just what I've been doing. Do what you want. It's going to be an exciting year, guys. Check out the Paul Saladino video that should come out tomorrow, Monday, and it's going to be an exciting year. I'm looking forward to moving forward with Emergence Diet Concepts. Glad to be back after a brief break. I'll see you guys next week.